Good afternoon, everybody. This is Abhilasha Daya, roll number 5, and I'm presenting to you the topic that's anatomy of lens. So let's proceed forward. Before that, I would like to introduce you some embryological part which is actually important. That is optical vesicles, like how it is formed basically. So it started with the optical vesicles. No, it's okay. So yeah, I deal with the optical vesicles, which is 25th day of gestation. Gestation it has been started. Then there is a formation of lens placard, that's on the 27th day of gestation, and then there is a formation of lens vesicle, that's on the 38th day of gestation, and then finally there is a formation of lens. So yeah, we'll again proceed with this only. So how it is actually forming? So it's a formation of lens vesicle on the 4th week. So optic vesicle includes the lens placard from the ectoderm. The most important question which actually asked from the PGs also, that's from where it gets originated, and that's the ectoderm. Lens placard invaginates and becomes the lens pit, and then the optic vesicle forms the ectoderm to become the lens vesicle, right? Now there is how the formation of lens fibers and zonules, which is the very most important part of this, to hold it into its place, that is primary lens fibers that forms up to the 3rd month. Cells in the posterior portion of the lens vesicle that elongate to fill the vesicle from the embryonic nucleus. Secondary lens fiber, that's from the 3rd month to the entire life they're going to form. So cells in the interior portion of the lens vesicle that divides actively. The most important here also, the question put up in further I'll proceeding, I'll let you to know about it more. And elongates includes all other nucleus, which is basically 4 parts. Lens capsule produced by the anterior epithelial cells. Lens zonules from the neuroectoderm in the ciliary area, which is from 3 to 5 months. So here, this is my topic as we know, anatomy of lens. So definition, first of all, foremost, we are going to, lens is the transparent biconvex crystalline structure. Position of lens in the eyeball, how it is being placed. So there is a place which is called patella fossa, which actually lies between the posterior surface of the iris and the vitreous in a saucer-shaped depression, which is called as a patella fossa, and there is a post surface of the lens. Now we are going to know the connectivity also. I have one picture, pictorial representation might be looking into this. So look into this thing, like what happens. So there is the posterior surface of the lens is in contact with the vitreous and attached to it in a circular area with the Vigert's ligament. Now there is a potential space between the posterior lens capsule and the Vigert's ligament, which is known as the Berger space or retrolental space also. So this pictorially you'll also come to know where these lies. This is the anterior hyaloid membrane which I talk about of the vitreous. This is how backwardly it is going to attach with the lens posterior capsule. There is a space of Berger or the retrolental space is not mentioned here. I am just verbally making you knowing that. So this is how. Now we move on to the functions of it. To maintain its own clarity, basically 3 most important functions of the lens we are dealing with. The most important is to maintain its own clarity or the transparency we are talking about, and to refraction of light, the foremost. The most important thing is to transmit the light rays and the focus onto retina and specifically onto the fovea part of it. Now, and the third thing is to provide accommodation. I told you that I'll make you know in future is this thing, that is your zonules, the fibers which latch it into this place. One question pops up in my head, that is, what happened? Why we are all discussing about this development kind of stuff from the embryonic to this accommodation and stuff? What happens if the development of the capsular part of the lens won't take place? Like what would happen? This question I'll end up in. If you're gonna ask me this question, I'll answer you. Otherwise, I'll have few questions on this topic too. And that is to provide accommodation. Okay, no blood supply or innervation after the fetal development. So again, there is a question then how it gets its nourishment and every other step, because it's not having any kind of supply, so it depends entirely on the aqueous humor. Now dimensions of lens, this is something very numerically and we have to know about this. Equatorial diameter of lens in adults is about 9 mm or 10, 9 to 10 you can say. There's something misprint over here. Now during the birth, approximately it's about 6.5 mm and it attains maximum diameters. Like it also furthers when when we are aging. So this actually further, like in the second decade of life I have mentioned. Axial diameter means I'm talking about the thickness of the capsule here basically. So at the birth it's 3.5 mm, and at extreme aging, when we are aging, so it's about 5 mm. Now the weight, it also varies, and different books have been written differently. I gotten is 135 mg from 0 to 9 years of age, and with the aging, the weight, because we all know the weight and the size of the lens during the aging, it increases. Because of that, it's all happening. Now the surfaces of lens, which is also an interesting thing to know here. Two surfaces, anterior and posterior, which are basically anterior surface and posterior. So whole it is being covered by the epithelium, but we divide it into anterior and posterior, anterior epithelium and the posterior epithelium. The anterior surface is less convex. So I would rather saying as a spherical, you would let it to know with an olive kind of structure, which is because it's something not very spherical, because the anterior surface is less convex, and it is a part of sphere having a radius of about 8 to 14 mm, and the posterior surface is more convex compared to that of the front one. That's the anterior, that's the convex, and is a part of sphere having a radius of 4.5 to 7.5 mm. Now we are talking about the poles of lens. So this is also anterior pole lies in the center. Whatever there is an anterior surface, anterior epithelium or anterior part of the lens, the midst of it, the center of it, that actually calls as an anterior pole, and the surface, and is about 3 mm from the back of the cornea. Now posterior pole, that lies in the center of the posterior surface. Okay. So there is again a function of it to just refract a light, that transmits a light, focuses onto the retina. That's what its function we are discussing so far. So the refractive index of the lenses, these mentions is what refractive index of lens is 1.39. In some textbook we are dealing it with the central refraction is more, peripheral reflection is less, that is about I think 1.04 and 1.36 from the periphery. So central most is the most one, and the refractive index of the second point is of the cortex, it means we are peripherally we are talking about, so 1.38, and the refractive index of the nucleus is 1.42. So refractive powers again, yeah, we have been doing it in the diopters, and it is also important, as I mentioned, accommodative powers, it varies with the age, which is actually important with the lots of refractive errors we are having, hypermetropia, myopia, astigmatism, presbyopias. So at birth it's very good, it's like 4 to 16 of diopters. At the midst of it, like on a 25, it's 7 to 8 diopters, and as we are going so far in the ages like 50s or beyond that, or mid 40s, or from 40 to 50, it's about 1 to something very less. Okay, now lens transparency. Now how it maintains itself to so transparent, so that the light, because if it is not transparent, light won't be able to transmit in midst of it, and you won't be able to just focus the picture onto the retina. So it's avascularity of the lens which aids into it. Single layer of epithelial cells it's having, semi-permeable nature of the lens capsule, okay, characteristic arrangement of the lens proteins, it's also very most important thing in this, it has. Here I am talking about the pump mechanism of the lens fiber, that is sodium potassium pump I am talking here. And actually we are very most important, like in anatomy of lens is also important to know its biochemistry, its physiology, because they are helping it to know, like there are lots of pump, there's lots of processes, glycolysis, glycogenesis, HMP shunts, and we have a sorbitol pathway that all aids in that. Okay, auto oxidation, when I talk about, it's also important. Now most important here you are looking is the glutathione, which is actually, I would say, it's a free radical scavenger. It's very important. And the lens protein in the reduced state, because it helps to maintain its clarity or its transparency, and provides the integrity of the cells to membrane pump. Now importance of refractive index again. If the refractive index of the lens is too low, just earlier also I mentioned why we are just discussing about the refractive index all the time, because it's important for the refractive errors we have been dealing in the lives, and that's, if it's too low, then it's called index hypermetropia. If the refractive index of the lens is too high, index myopia. In older ages, the refractive index of the cortex relatively increases and approximates that of the nucleus, and lens converging power, I become hypermetropic. And cataract I mentioned very shortly, although it's a very important, a very biggest and the vast topic to discuss all alonely. But here I need to mention, because any pathological thing happens or any refractive errors, it all lead to the cataract. We have a different variety, and I know everybody knowing that. Become myopic, okay, increasing the refractive index. Just to take a glance on it, I just mentioned it here: diabetes, hyperglycemia, increase in the blood glucose level, or diabetes. Why we mention here? Because again I told you the biochemistry and the physiology are very much concerned about this topic too, as there are a lot of more pathways to let it to maintain and sets its own functions, it's on everything. So here is the diabetes. Why? Because there is increase in blood sugar level. So what will happen? There is glycogenesis, everybody knows that pathways. When there is an excess of glucose, then there is a negative feedback, right? And then the glucose goes to the sorbitol pathway. So it means finally what we are doing, that glucose is converting into sorbitol, another kind of glucose, and then further it emits as in the form of fructose, which actually sits into the nucleus. We don't give get an escape out from that nucleus. It sits there. So because of that, water draws in, it swells up, breaking of the fibers and a lot more stuff in the lens, again lead to cataract. So thereby there is an importance of this. So now we are dealing with the part of lenses parts, basically three most important: capsule, then we deal with the epithelium, then we have the fibers. So the first and the foremost is the capsule. So elastic transparent membrane capsule as we are having, it's actually made from the type 4 collagen. It doesn't mean that the cell or the lens of our eye is only made from this kind of collagen. We are also made from the type 1, type 2, type 3. But basically when we stick onto the capsule, it actually emphasizes on the 4th type collagen. Thicker anterior. Now what happened? I told there's an anterior part of the capsule, there's a posterior part of the capsule. So when this capsule, it surrounds whole of your lens, so the anterior part of it is termed as an anterior capsule, and the posterior one term named it as a posterior capsule. Now here is the point, that the anterior one is the thickest and the posterior one is the thinnest one, posterior pole. And why is it so? That anterior, that I'll let you know further. Now the thickest also, the most important question I found it also, that it's been asked also, that what is the thickest membrane, basement membrane of the body? Hence this is your answer, that's the capsule too. Now light microscopy appears as a homogeneous structure, but electron microscopy shows you the lamella bed. This is how we're going to see onto the microscopic levels how it beautifully can be expressed. Zonulars zonulars mean I'm talking about the fibers here, get attached to at it at the equator. It has two prospects of attachments, like also in the fibers I'll let you know, but some part I want to let you know here, that zonulars attachment to the equator, like how this is placed in the position. So when it is from the equator, it's 1.25 mm, 1.5 mm. From the equatorial posterior to its 1.25 mm, this is how it is get attached to it, and it's hold itself there. Now we are having here zonular get attached, that I let you know just now. Age related loss of accommodation, yeah, this is a function of lens. Hence here, as I told you, as we are aging, a lot of complexities we are dealing with this. So age related loss of accommodation results from the change in capsular elasticity, which may be due to the loss of lamella. Okay. Now here I mentioned is anterior epithelium, but basically the topic is epithelium, but this is the foremost important, so I mentioned it into a heading of anterior epithelium. So it's a single layer of cuboidal nucleated epithelial cells. It is most important because posterior epithelial or the posterior epithelium, if I talk about, it's very thin, it's didn't like the one it is having here, 5 lakh cells in the mature lens. Yes ma'am, only a few slides are left. Now the only metabolically active part of the lens is only this. Now the density increases towards its periphery, and now no posterior epithelium, as this is used up in the formation of primary lens fibers, which occupies center of the nucleus. Now basically we have the three zones. I need to take a lot more time to just let you know in more deeply, but I'm just mentioning, finish it off here. Central, intermediate and terminal. I'm so sorry about it. This is a picture. I think it's not clear much, but you have to just be aware of this. This is the most important. Here is also a foremost question. I didn't put it up because of the time limitation. Those reasons, it's a very most important question from here. And this is the transitional zone. This is the active mitotic region I just talked about. This is germinative zone. This is the equatorial. So there's an immigration of cells, and there, and then again there's a posterior. A lot I need to explain, but I cannot. Sorry. So let's move on to fibers. Now epithelial cells elongate to form the lens fibers. At first, lens fibers are formed from the posterior epithelium, later to form the equatorial region of the epithelium. Now, the cytoplasm of the cells of newly formed fibers contain the rich ribosome indicated elevated protein synthesis. This is also again very important, but I can't express here. Sorry. Now the nucleus disappears later on. Now there is a ball and socket. Important here. Like I'll finish it into my words because it's too late. So yeah, what happened? These fibers get into attachment with the Y-shaped structures. Why the question is Y-shaped? Because they are attaching into a 3 dimensions initially. That's why we termed it as a Y. Anterior one attach in the form of anterior Y. Posterior ones when are attaching towards the sutural we are talking about. That's innervated Y. Or you can say inverted. But later on we can't use these words because later on when they are combining together they are forming into a different dimensional structures like 9, 12, so they are not able to form this kind of shape, but this is important here. The later stages, the lens sutures arranged in a complicated dendritic pattern. Here I mentioned last point. Nucleus. Most important, the nucleus contains the oldest fibers as we know, and the cortex is the newest one, again question. Nucleus always contains the older fibers, newer one formed is the cortex one. The embryonic nucleus forms between 1 to 3 months. Infantile nucleus formed from birth to puberty. Then adult nucleus, there are 4 types, then correspond to the adult life. This is somewhat a picture. Pictorially I want to mention you here. This is how you can see that. Now cortex, again it's a peripheral part of that lies just outside the nucleus. Adult nucleus, and this form the youngest fibers. Okay, now ciliary zonules again important. A series of fibers which hold the lens in position and enables the ciliary muscles to act on the lens. Made up of fibrillin with the larger glycoprotein. I'm sorry, I again have one important you to know about here. If this fibrillin protein from which these zonule fibers have been formed, if there is mutation in this, so what will happen? I'll ask you at the end of this. Composed of microfibers with a diameter of 8 to 40 nanometer, transparent, stiff and non-elastic. So yeah, I end up here with this my slide. That's the biochemical composition. Very shortly I mentioned it here. 65% of the water and 34% of the protein. That doesn't mean here it contains the lesser of the protein, because comparative to the other part of the body, this is the only part of the body which contains the higher percentage of the protein, which is important. Again, aging, cataract, everything concerned with this topic. The lipid, carbohydrate phenomenon, ascorbic acid again, glutathione, the most important, amino acids, and the inorganic.